The effects of carbon dioxide on the rate of photosynthesis. The purpose of this lab is to determine the effects on the rate of photosynthesis in varying concentrations of carbon dioxide by observing the decrease in density of the plant disc due to the production of oxygen. To do this, we will be observing the rates of which oxygen gas is released by leaf discs submerged in various concentrations of sodium bicarbonate solution. Hypothesis: if there is an increased level in carbon dioxide concentrations in the external environment, then the rate of photosynthesis will increase until a saturation point is reached. This is because the higher the concentration of CO2 is, the more likely effective collisions between rubisco and CO2 are to occur. However, once all enzymes are used, then the increase in CO2 will not increase the rate because the enzymes are all saturated and a plateau will occur. The things you'll need will be baking soda, distilled water, a weigh boat, a scale, a pipette, 10 milliliters of liquid dish soap, a syringe, a hole puncher, a few philodendron leaves, a 400 milliliter beaker, one 300 milliliter beaker, 3 250 milliliter beakers, a timer, and a light source. We'll start with creating a liquid dish soap solution by combining 5 drops of liquid dish soap and 5 drops of water. Next, we will be preparing the 0.2% solution of sodium bicarbonate by placing 1.39 grams of baking soda into 300 milliliters of water. In addition to that, we'll be adding 1 drop of diluted liquid dish soap. Adding this soap will increase the permeability of the leaf because leaves have cuticles which are hydrophobic. The soap will break down the cuticle layer and allow the solution to enter the leaf. To make the 0.4% solution, repeat the steps from before, except this time add 2.6 grams of baking soda. Using the hole punch, cut 10 uniform leaf discs for each of the 6 trials. Try avoiding any major veins of the leaf. The next step is to suspend the leaf disc in the 0.2% solution. This is a very important step because the leaf disc has oxygen in it, so it will float. So to make them sink, we will be removing the oxygen and pushing in the sodium bicarbonate solution so that there's also a source of CO2 for photosynthesis to occur. Remove the plunger from the syringe and place 10 leaf discs into it. Carefully reinsert the plunger to avoid crushing the leaf. Push on the plunger to remove excess air until the leaf discs remain. Leave about 1 millimeter of airspace to avoid crushing the leaves. Next, extract 20 milliliters of 0.2% solution of sodium bicarbonate using the syringe. Create a vacuum in the syringe by placing a finger over the opening and pulling back on the plunger. Hold this position for about 10 seconds. While maintaining the vacuum, move your hand in a circular motion to swirl the leaf discs and suspend them in the solution. Let go of the syringe to release the vacuum. Repeat this step 2 to 3 times until all the discs sink properly. If they don't sink, add 3 drops of soap into the sodium bicarbonate solution and repeat steps 7 to 10 until all the discs sink. Be careful not to overdo this step because over-suspending may cause damage to the leaf disc and affect your results. Once all the discs have sunk into the bottom, remove the plunger from the syringe and carefully pour its contents into an empty 250 milliliter beaker. Then add water until it reaches the 100 milliliter mark. Immediately after, place the beaker under a light source and start the timer. Record the time for whenever a disc floats to the top, and occasionally swirl the beaker around to dislodge any that might be stuck up against the sides of the beaker. Continue to record until all of the discs have floated to the top. One of the things to note here at this point is to watch the time between suspension of the leaf disc and transferring them to the different solutions. This is a source of experimental error because the syringes are not left in the dark and photosynthesis is still able to occur while the leaf discs are still in the syringe. This is the reason why the first disc of several trials floated to the top in less than 1 minute from being transferred to the beaker. Once everything is done, conduct a second trial and average results of the two. That will give you the results for your controlled experiment. Then, repeat steps 1 to 12 again, except this time, instead of topping off the beaker to the 100 milliliter mark with water, top it off with 0.2% bicarbonate solution. After conducting 2 trials with 0.2 bicarbonate solution, repeat steps 1 to 12 once again, except this time top off your beaker with 0.4% bicarbonate solution and record your observations. When the beaker is placed under a light source, the light-independent reactions of photosynthesis will occur. Photons will get excited and the Z proteins will split molecules of water, releasing oxygen gas into the air spaces of the leaf disc. At the same time, the carbon dioxide will be used in cellular respiration to make glucose, and the leaf disc will start to float. The production of oxygen and time it takes for the disc to float is how we will determine the rate of photosynthesis. And here are the results of the experiment. As seen in the graph, the results contradict the hypothesis. The rate of the controlled experiment was significantly faster than the rate of the photosynthesis that occurred in the 0.2% solution, and the rate in the higher concentration bicarbonate was actually slightly slower than the rate of the controlled experiment. This result was caused by the combination of human and experimental error, as well as the design of the lab itself. One of the first things to know is the impact of the previously mentioned error. As seen in Figure 1, the first leaf disc to float for the 0.4% solution on average floated to the top just after 30 seconds of being placed into the beaker and placed into the light. This is the fault of multitasking and suspending the leaf disc too early, as well as exposing them to light for about 4 minutes before transferring them. Another source of error that greatly impacted the results was the different light intensities that each beaker was exposed to. As seen in this image, the beaker that was the controlled experiment was actually elevated in comparison to the beaker with the 0.2% solution, meaning the beaker that was the control actually was exposed to more intense light. And sadly, light intensity is a factor that affects the rate of photosynthesis when a plant has yet to reach its light saturation point, which includes the light-dependent reactions which we were measuring to monitor the amount of oxygen that was released that allowed for each leaf disc to float. Therefore, the leaf disc in the controlled beaker would have received more light and therefore would have created more oxygen, allowing for the leaf disc to float significantly faster than those in the 0.2% solution beaker, which explains why the results show a slower rate of photosynthesis in the beaker with the 0.2% sodium bicarbonate solution, because before a plant reaches its light saturation point, a higher concentration of CO2 will not increase the rate of photosynthesis in that plant. CO2 is only a factor during light-independent reactions with regards to enzyme concentrations in the Calvin cycle. This experiment should have happened in a control environment where light intensity was the same for all of the trials to give accurate results. Which just goes to show some of the obstacles people face when designing a lab, which includes, but is not limited to, having a very good understanding of external variables that may alter the results of the lab, being very precise, and making sure that the procedure will be fair and is designed to give the most precise and accurate results, and most importantly, proofreading and understanding the significance of each and every step of the experiment and its impacts to the result. For this lab in particular, one obstacle that was faced was that we overlooked the importance of the solution the leaf discs were suspended in. We were expecting to have 0 leaf disc float in our control experiment and were unpleasantly surprised when more than one did. So really, for that experiment, it should have been suspended in just water and soap.